Okay, good morning everyone. Yeah. How are you all doing? Good? Okay. So today we are going to talk little more detail on antigen presentation and know more about, in general actually, how antigens are processed and presented, what cell types are involved, what are the different processes which are critical in that event. Because tumor antigens actually the same thing happens. So this will be a very general discussion. There will be some tumor reference, but it will be mostly in general talking about dendritic cells, macrophages, co-stimulatory molecules, antigen presentation, CD4, CD8, and all those everything and in between. So antigen presentation is basically a process where antigens are basically presented at the cell surface, which will be recognized by lymphocytes. And the process in itself involves antigen uptake, degradation and processing of that particular peptide, and then finally antigen presentation. And I think from day one, I'm keep hammering you guys with antigen, and antigen. This is a classical definition of antigen, a molecule which reacts with preformed antibody and or specific receptors on T and B cells. Okay, and those receptors will be your T cell receptor and B cell receptors. So I will give you a brief introduction also about those two receptors. So what are the requirements for antigen presentation? Of course, you have to have these antigen presenting cells. And what cells do antigen presentation? That not everyone. You know, there are a few of them that do it. Can you give me a name of one of them? Macrophages. Everybody loves macrophages, seems like, you know. The title of the talk was dendritic cells, and you forget dendritic cells. Actually, you know, sometimes your answer has to be in priority, okay? There are three cell types which basically are involved in antigen presentation. Your dendritic cells, macrophages, and B cells. And if you are asked this question that you have to pick one antigen presenting cells, okay, you have to go with the probability, you know, that if all three are there listed in your multiple choice question, you cannot just pick all three, okay? So I think and if somebody asks you, give me a name of antigen presenting cells, I will just go with dendritic cells, you know, because that's their job. That's the, you know, I will show you why macrophages they don't exactly come to that level of antigen presentation, because macrophages basically needs help. They need help in order for them to become fully functional antigen presenting cells. Antigen presenting cells and T cell trafficking, again, they have to come together. Once the antigen is picked up at site of infection or injury, they basically travel, and which tissue organ this interaction happens, APC T cell interaction, where it happens? At the site of injury or infection, or where? Secondary lymphoid organ, lymph node, spleen. Okay, so those are the primary site for these antigen presenting cells and T cell interaction. T cell and B cell receptors, definitely these are required. There are many co-receptors which are involved in this process, and of course co-stimulatory molecules are part of the regimen. So there are a lot of specific requirements for antigen presentation, and you will come to realize at the end of the talk that why they have to have so many multiple controls, you know, checks and balances in order for it. And the basic idea is that to have a very defined, specific antigen dependent response. So antigen presenting cells are actually present throughout our body. Within secondary lymphoid organs, they are not distributed everywhere. If you look at macrophages distribution in a lymph node, they will be everywhere. But if you look at the location of dendritic cells, they will be only in cortical region and medullary region, not in the follicular region. And the same thing goes for B cell. You don't find B cell anywhere but in the follicular zone of a lymph node. So they are basically localized into specific areas, differentially distributed. These cells are dendritic cells, macrophages, and B cells. These are the only cell types. They are capable of expressing co-stimulatory molecule, and when suitably activated, they can activate naive T cells. So there are two specific requirements for these cells. One, as I said, co-stimulatory molecules. These are the only cell type which expresses co-stimulatory molecule. What will be the other requirement for antigen presenting cells? Wake up. Tea break will be 1 hour away, okay? I'm sure you had good coffee. MHC class 2 molecule. MHC class 2 molecule, all antigen presenting cells will have to express. It's restricted to these 3 cell types. And MHC class 1 is expressed by all cell type in our body. So here is a very generalized view of antigen presentation, processing, and T cell activation. You have antigen, which could be anything. That antigen will be picked up by immature dendritic cells, and as it is picking it up and processing, it goes through the process of maturation. While it is traveling, also it gets activated, and then it will go to the secondary lymphoid organ, like your lymph node or spleen, depending on where the infection or injury happens. Over there, these dendritic cells will present their antigen to naive T cells, and these naive T cells or B cells, depending on the type of antigen, again, can generate your antibody producing plasma cells or produce cytotoxic T lymphocytes, activate CD4 positive T cells, and try to resolve these infection, injury, or remove those antigen. And also in this process, they also generate some memory T cells. So the next time, whenever that kind of exposure ever happens, they don't have to go through this whole boot camp. Those memory T cells and memory B cells in itself can get help and can they proliferate and become more effector cells, and will take care of it. There is a whole bunch of innate immune cells in this picture. These are independent of antigen. They are the ones first reach over there whenever there is a tissue or injury, and they create these inflammatory milieu. And some of the cytokines or factors which are produced during that inflammatory response are important for driving maturation of these immature dendritic cells or macrophages to become fully functional antigen presenting cells. So here is a simple cartoon showing you different antigen presenting cells and where they are located. Dendritic cells, of course, they look like this, so that's why they are called dendritic cells. And if in a lymph node, you can see they are everywhere except they're in medullary region. This is cortical region, and these are follicles actually. This is where your majority of B cells and T cells reside. In case of macrophages, they are everywhere, and in B cells, majority of them, they are localized into these specialized area, they call it follicle. There is some specificity that what kind of antigens these 3 different antigen processing cells are involved in. Most of the viral infections, definitely it is initiated and processed through your dendritic cells. Bacteria, definitely macrophages, and some of the toxins and other antigens which are not processed through these, they can get to the B cells. One key difference between these 3 cell type, you can see in these 2, these antigens, once they are taken up, they have to be processed, and then presented in association with class 2 MHC class 2 molecule. In case of B cells, there is no processing. Okay, that antigen, if they have that particular antibody, that antigen will bind to that B cell receptor, which can recognize that antigen, and then that bound B cell receptor will interact with CD4 positive T cells, will drive the activation and differentiation of that B cell into plasma cells, and you have antibody. Okay, so there are very distinct pattern and activities which are assigned to all these three different antigen presenting cells. So dendritic cells, macrophages, and B cells, they all differ in their cell activity to antigen processing properties and their co-stimulatory molecule expression and migratory behavior. Thus, they have a very distinctive function in initiating T cell response, and it's all driven by the type of antigens is presented to them. In this little complex cartoon, there are a lot of informations which have been included. You can see these three different type of antigen presenting cells, and what are the specific characteristics when it comes to antigen uptake. Dendritic cells are pretty good at it. They can take these antigens in multiple ways. Macropinocytosis, phagocytosis are tissue dendritic cells through viral infection. Macrophages are good at eating everything as phagocytosis, and in case of B cells, these antigens basically bind to this B cell receptor because they are unique and they can initiate the response. When it comes to MHC expression, there is a low level MHC expression always in dendritic cells, so you can find these cells are always ready to go. In case of macrophages, it has to be induced by uptake of these bacterials and production of specific inflammatory cytokines, and I will show you why we require adjuvant in that context, because macrophages in itself, they are always not ready for antigen processing or presentation. In case of B cells, they don't need any activation. Our MHC class II is always expressed, constitutively expressed in these cells. Co-stimulatory molecule expression, basal level is always expressed constitutively in these cells, whereas in macrophages it is inducible. Basic unactivated macrophages, they don't express these co-stimulatory molecules, but when they interact with this bacteria, they phagocytose them. Inflammatory cytokines like TNF, interferon gamma, and all those cytokines, they activate co-stimulatory molecule expression and macrophages, and then you will have antigen presentation and T cell response. In B cell case also it is inducible. Type of antigens which are presented again are also little different. In case of dendritic cells, they are professional antigen presenting cells, so basically they can do everything, but they are predominantly involved in processing peptides, viral antigen, and allergen, and also intracellular threat. Okay, anything which is coming from inside the cell, intracellular threat. Macrophages, on the other hand, they are very good at processing and presenting particulate antigens, intracellular as well as extracellular pathogens. And for B cells, that antigen has to be free floating. They don't need to be processed. That all they have to do is that they have to be recognized and bound to these antigen binding domain on the B cell receptor. The location, dendritic cells you can find everywhere. Lymphoid tissue, connective tissue, and many epithelial linings have dendritic cells present. Macrophages are localized in lymphoid tissue, in circulation, in connective tissue, and most of our body cavities. And B cells, of course, peripheral blood and lymphoid tissue. So you can see there are, although they are all antigen presenting cells, they have different characteristic features and a different job actually to do. When we say intracellular threat, basically what you're talking about is that if a cell gets virally infected or if cell is transformed, that cell basically has to be recognized and removed. So any epitope or antigen which is getting presented in that needs to be processed. And in that context, your majority of the time, your dendritic cells come into the picture. So dendritic cells are professional antigen presenting cells. Usually, once they pick up the antigen, which are released through initial assault of our innate immune cells, or the inflammatory milieu which is getting created in the initial phase. In that process, antigens are released, and those antigens get picked up by your dendritic cells, macrophages. In this context, we are talking dendritic cells. They migrate to lymph node, and as they are migrating, they get activated and mature. At that time, they also overexpress these co-stimulatory molecules. One key element to all of these activities is innate immune response. Without inflammation, nothing happens. So yesterday we talked about inflammation is good and bad, and good in the sense that without inflammation, we will never have an effective immune response. Inflammation is important right from the release of the antigens to the activation of these antigen presenting cells, and effectively presenting it to naive T cells and generating effector T cell population. So here is a very simple cartoon. You can see immature dendritic cells in peripheral tissue. These cells actually, when they are at a basal level, they do express different molecules which are important and interesting in terms of taking up the antigen, not so much in the presentation. As they go through and try to reach to this lymphoid tissue, there are few things which show up, as they are maturing. One thing definitely is this chemokine receptor, because that's what drives them to the lymph node. So CCR7 gets up, and the ligand for this CCR7 is CCL20, and that has been utilized clinically now to bring more dendritic cells in tumor. And I will talk about it during our discussion, what the useful things they are doing in that. The other thing you will see is that co-stimulatory molecules expression starts to show up as they mature. This is BC1, and the two popular ones are B7.1 and B7.1 and B7.2. And also it upregulates MSC class II expression. It has some basal level of MSC class II expression, but you will see that MSC class I goes down and MSC class II expression goes up in these cells. So there are specific cellular and some of the adhesion molecules also, start to show up in a higher affinity compared to what they had early on. So dendritic cells mature through at least in two definable state, which becomes very potent antigen presenting cells as they are going through as they are traveling through the lymphatics to the secondary lymphoid organ. So naive T cells basically come into the secondary lymphoid organ. Best example is your lymph node. They are sampling this MHC peptide complex as they are migrating through the secondary lymphoid organ. This is one important, take home message. Most of the cells, once they get out, at least T cells, when they get out of thymus, they are naive. They have all the machinery, they know where to go, but they are not effective. In order for them to be effective, they have to be, presented through the antigen in context with MSC class II molecule, and then they will become active. And all these processes are happening in secondary lymphoid organ. So here is a simple cartoon showing the T cell trafficking, and as they are trafficking, they are interacting with dendritic cells or antigen presenting cells and getting activated. So I will walk you through T cells. They enter into the lymph node through high endothelial venule. There are two ports, efferent and efferent, so they enter through the efferent. And these naive T cells basically, they will come in lymph nodes. Dendritic cells are already over there. They have already are ready to present their antigen. These T cells will interact with antigen-presenting cells, either macrophages or dendritic cells, depending on the type of antigen. And you can see here they are just bound together, they are interacting with, and your antigen is getting presented to these naive T cells. And at this stage, these cells, what happens is that these naive T cells, now they know which antigen they need to target, and there is another activation and proliferation phase where they start to multiply. And one cytokine which is really getting upregulated at this moment is called interleukin-2, which is important for T cell proliferation. And there will be a clonal growth of those T cells. So many different, many of those will be produced, and that basically once they multiply, they proliferate, and these armed effector T cells basically will get out of the lymph node and go and do their job. And at this stage, what happens to these antigen-presenting cells? Most of them undergo apoptosis. Once the job is done, these dendritic cells or macrophages, they undergo apoptosis, and they have to be eliminated, because you don't want to have this continuous antigen presentation of the same type of antigen, because then that will lead to more pathological conditions. So if we summarize what we just showed, is naive T cells, usually they encounter antigens during their circulation as they are going through the secondary lymphoid organ. Those T cells that do not encounter any specific antigen receive a survival signal through their interaction with self-MHC molecule or cell peptide and leave the lymph node. This is a very important point actually, because those you have a very defined number of T cells, okay, a T cell repertoire we call it, okay. So those naive T cells, you don't want them to be eliminated. You want them to be surviving and thriving. So basically those which they don't see what the antigen they need to bind, they will get the survival signal, and that survival signal is your self-MHC cell peptide complexes which are there. Those actually which will interact and encounter the specific antigen will get activated, proliferate, and differentiate into armed effector T cells. And these T cells basically will leave, and these T cells which know exactly which antigen they need to target will go to the site of infection or injury and do their job. The one key point here is that these T cells, effector T cells, later on they also lead to, some of them will define and differentiate into memory T cells, which is very interesting. The question here is that whenever we are talking about why few T cells will encounter these antigen presenting cells, majority of them will not. And it all depends on the type of antigen, okay? What antigen they need to encounter. And who decides and where is the selectivity comes. I think I have mentioned a couple of times that each T cell has a unique T cell receptor, or B cell have a B cell receptor. So if antigen X is there, which is getting presented, in that context, any T cell which can recognize that X will interact with that dendritic cells, and everything else will not, okay? So very selected, or one clone of that T cell population basically will interact at one time point, and rest of them, they will just get a survival signal and then leave. So I will just show you in few slides about how these T cell receptor or B cell receptor look like, because they are very important in our adaptive immune response in context with antigen. So here is a very simple cartoon of T cell receptor. It's a heterodimer protein. Majority of cells, T cells in normal human being, they are alpha beta T cells. Very few cells, T cells are gamma delta T cells. And if you look at, this is a very simplified version. This is a structure of antibody molecule. You have an antibody molecule is basically made from 2 light chain and 2 heavy chain. These are light chain and these are heavy chain. And in that, they create this complex where this portion is responsible for antigen binding, and this portion is responsible for effector function. So every antibody is bifunctional. One portion of the antibody usually is important for antigen recognition, other one is for effector function. And effector function is basically, I think I mentioned you yesterday that these Fc portion will go and bind to Fc receptor expressing cells, and that antigen antibody complex will be eliminated, or the cells will be killed through antibody dependent cellular cytotoxicity. So in case of T cell receptor, basically this portion is what you see is here, which is a B alpha variable chain and constant chain, and this is what is important for antigen recognition. So structurally, this FAB portion of an antibody is what it looks like, a T cell receptor. Okay? And in case of B cell receptor, what is B cell receptor? Anybody? Immunoglobulin molecule, either it is IgM single chain or IgD. So basically your B cell receptor will be like this. Okay. Anyway, let's get with the T cell receptor first, and I will show you B cell receptor also. So T cell receptor is a disulfide linked heterodimer protein, alpha beta or gamma delta T cell receptor. T cell receptor enables T cell to recognize a diverse array of antigens, and it is associated with a complex of polypeptide. They are collectively called CD3, and this is what you use to stain T cell. All T cells in our body, they express CD3. Okay. This is a B cell receptor. Again, it is a monomer of either IgM or IgD. They consist of 2 identical heavy chains and 2 light chains. So this is basically your FAB portion of the antibody, and basically looks like it is inserted into the cell membrane, B cell. So this is what the structure of B cell receptor looks like. The main component is your monomer of IgM or IgD, and it has also some associated complexes. Basically, they are called secondary component or accessory molecule. They are critical in downstream signaling for B cell maturation and differentiation once the antigen is recognized, and they get help from CD4 positive T cells. So I told you that in normal physiology, or at least we all have T cells and B cells with T cell receptor or B cell receptor capable of recognizing any and every possible antigen we will ever expose to. Okay? And that comes through a very defined process called T cell diversity, TCR or BCR diversity, and usually it is generated through a process, a recombination process. Okay? And in this case, it will be a somatic recombination, right? Yes. So there are like multiple genes which make one T cell receptor. Okay? I showed you alpha beta is a heterodimer protein, so at least you will have alpha portion and beta portion. Okay? Even in alpha portion, you will have multiple genes encoding from alpha. Okay? Long peptide chain. So here, that's what it is saying, is that it is generated by 4 different genes. Alpha and beta genes are expressed on majority of T cells. Even with alpha, you have this variable region, diversity region, and joining region, and there is a constant region. And a combination of all these gene portions, actually, are domains in another word, will create a new variety of T cell receptor. Okay? So basically, what I will tell you is that with these, and again, another variability you have, that there are multiple variable region genes. So it's not one. Somewhere between 5 to 250 different variable region genes are there. Same is for diversity, and same is for joining region. And there are many other minor variations which gets created. So think about it, in a simple way, like how many of you are in this class? Okay, 30, okay? 30 of you are in this class. And if you have to make a, group of 3, okay? How many different groups you can create? And you can switch back and forth, possible groups. You don't have to stay in the same group, okay? How many possible groups you can create? 30 students, 3 per group. How many possible combination? I don't want you to have T, just 3 of you together. And just think about it, how many combinations you can create. It's simple mathematics. 33 per group. 10? 10 groups? So how many groups? I'm not going forward, okay? Because this is important. If you understand this, you will understand, how many different, combinations that can be created, okay? It's a simple mathematical model. So how you do that? Speak loudly. I cannot hear. I said I am old now. 3 raised to 30. That's the right answer. Okay, that many different combinations you can create. Okay, and so this is what happens. Okay, this is the very busy chart, but all you need to know is that for this is for your B cell receptor, and this is for T cell receptor alpha beta and T cell receptor gamma delta. There are many variable genes, which encode these sequences. There are many diversity genes, and also there are variability in the open reading frame of these diversity gene, joining gene, and there are some nucleotide substitutions which happen along with these. Same thing goes for the variable region. There are minor changes. All in all, this number is what you need to remember, okay? That at the end of the day, there are 10 to the power 11 different type of B cell receptor expressing B cells, okay, there, okay? It's just a guess, okay, based on this. For alpha beta T cell receptor, 10 to the power 15, okay? And for gamma delta T cell receptor, you are looking into somewhere 10 to the power 18 different possibilities, okay? And I don't think you will ever encounter these many antigens. In our lifetime, we will not, okay? Another important thing, besides this diversity, what it is allowing is that basically, of course, it is equipping us to defend us from any and every infection. And in case of T cells, majority of these T cells with specific T cell receptor alpha, beta, gamma, delta, they are all done by the time we become adult, right? Do you know why? Once we become adult, there are no more new T cell things. Yeah, thymus basically undergoes atrophy at adolescence. It is basically no more new T cells are getting, basically trained. They are generated, but they die, okay, because they cannot go through the process. B cells, they continue to throughout our body, throughout our lifetime, Actually, this is again not a textbook example, but for those patients who undergo these high-dose chemotherapy and stem cell transplantation, in those patients, we have seen extrathymic, T cell, maturation. In that case, basically, that they are adult, their thymus is either getting rejuvenated, or there are other organ and tissues, where these T cells are getting, processed. And these are the cells, basically, you are infusing them, you are giving them these, stem cells, which have hematopoietic, progenitor cells, which give rise to all your T cells, B cells, and other cell type. So we have seen, even in those adults who are getting these stem cell transplant, there is extrathymic, T cell maturation and processing happening. So but that's not a textbook example. In textbook, basically, you have enough T cells, different type of T cells, a T cell repertoire, which are capable of defending you throughout your life. So a T cell recognizes antigen in the form of a complex, a foreign peptide bound to an MHC molecule. And this is, of course, I think you all know about by this time, that they have to be bound to either MSC class 1 or MSC class 2. MSC class 1 definitely is in all cell types. MSC class 2 is only in the antigen presenting cells. But in case of B cells, they don't require that. B cells, basically, these B cell receptor directly can bind to antigen through that antigen binding domain, and can generate and get differentiated and get, produce more antibodies. All alone, these T cell receptor complexes, they don't contain any signal motif. I will show you the structure. These T cell receptor CD3 complexes are always in association with CD4 or CD8 molecules, which is very critical for antigen specific signaling. And one thing you will notice is that these CD4 and CD8 molecules are important in interaction for MHC molecules. So here is a very simple cartoon of a T cell receptor CD3 complex. You have T cell receptor alpha beta here, and this is a CD4 molecule, or this is CD8. This is a you cannot, in most adult mature T cells, you will not have CD4 positive or CD8 positive T cells together, okay? Either there will be CD4 positive or CD8 positive. But if you look in the thymus, those T cells, to begin with, they are both double positive. They call it CD4 positive and CD8 positive. But in mature in peripheral blood, you will not find it. So either they are associated with CD4 or CD8. And in this CD3, basically, is this complex. These four polypeptide chains, they form this CD3 complex. The most important molecule in this picture is this zeta chain. This zeta chain is what is required for downstream signaling. And actually, that has been exploited in generating some of the chimeras, where actually they have just antigen binding domain tagged to this zeta complex and can be utilized as a factor T cell moiety. And that is in very much clinical, preclinical stage. So T cells with the different functions, they have either associated with CD4 or CD8. They recognize antigen or peptide in a different class of MSC molecule. CD4 usually interact with MSC class 2. CD8 usually interacts with MSC class 1. And CD4 and CD8 are also called as a co-receptor in this TCR complex. So here is what CD4 and CD8 distinct picture looks like. This is CD8 T cell receptor, and in this context, you can see these CD8 positive T cells. Most of the time, they are cytotoxic T cells. They recognize these antigens which are presented in context with MSC class 1 molecule. There is a dual recognition going on here. Your T cell receptor, basically, it recognizes this antigen or peptide, which is over here. And another recognition is going on is with this CD8 molecule, which is sampling this MHC molecule for self-MHC, So there is a dual recognition which goes on in both cases, actually. The only distinct only distinction we can find in context with CD8 or CD4 is the type of MSC molecules which are involved. In this case, it is MSC class 1 molecule. In this case, it is MSC class 2 molecule. Okay, and actually, you can, by looking at the type of antigen or type of insult or infection, you can predict what kind of cells will get involved. As I said, that in case of most virally infected cells, where these viruses get inside the cell, they get integrated in the genome, or start producing some unique proteins. These proteins will be processed and presented in context with MSC class 1 molecule. Those antigens which will be picked up by your antigen presenting cells like dendritic cells are mostly macrophages, those will be presented to the T cells in context with MSC class 2 molecule, and that will be happening through CD4 positive T cells. So you can see, which cell type will be involved in what kind of threat. In most intrinsic, threats, you are looking into involvement of MSC class 1 CD8 pathway, and in most extrinsic type of threat, you are looking into MSC class 2 CD4 positive T cell. So CD4 TCR interaction with MSC class 2 peptide complexes are important, and actually what it does is that it also increases or activates these cells many fold. That recognition or interaction with MSC class 2 or class 1 molecule, it potentiates the whole process in manifold. So your T cells can respond better even if there is not enough antigens are present. In case of CD8 positive TCR complexes, exactly the same thing, only difference is that in this context you are looking into MSC class 1 molecule. Stable binding of protein peptide to MSC molecule provides effective antigen presentation at the cell surface. Both MSC class 1 and class 2 molecules effectively be prevented from acquiring peptides from surrounding extracellular fluid. Okay. This is a unique feature which prevents some abnormal activation, because these MSC molecules, and the complex gets together, they cannot just pick up antigens from everywhere. Okay. These antigens, when they have to present only those which gets processed through the intracellular processing machinery, and which is a very defined process actually if you go and look into. So they are incapable of presenting or acquiring the peptides or antigen from the surrounding extracellular fluid. If they have to do it, what exactly they need to do in order to do that? If there are some antigens which are present in that particular organ or tissue which are outside, not associated with MSC class 2 or class 1 molecule, what exactly these cells need to do to take care of those antigens? Yes, so those antigens, they need to be again picked up by any antigen presenting cells. Most probably it will be macrophages, because they are capable of eating and drinking, and they are in every tissue. So they will take it up and they will process it. Okay, they cannot just go and bind to MSC class. They have to be processed, and again they will be presented. And that basically is very much spares attacking or destroying the surrounding tissue, because during the processing of these antigens, if they are not worthy enough to be presented, these cells will never present those antigens. If they don't need to be presented, they will not present it. During these antigen presenting cells and T cell interaction, one important set of molecules which are really important is called cell adhesion molecules. These two cells have to come together, and that is mediated through a specific type of cell adhesion molecules, and most of them actually are coming from these immunoglobulin superfamily members, your LFA1 and other selectins. There is a transient binding of these naive T cells with antigen presenting cells, and that basically brings those complexes together physically. When naive T cells recognizes its peptide MSC ligand, signaling happens through the TCR complex, which causes a conformational change in adhesion molecule to stabilize these antigen presenting T cell complexes. And actually, this synapse which happens between these antigen-presenting cells and T cells is still area of exploration. There are a lot of things going on. Still, we are not the physical interaction and all those adhesion molecules. Those are all well characterized and well studied. But there is something beyond going on, because the responses which they get for specific antigens are very diverse and different depending on the type of antigen. So that synapse is another hot area of research and trying to manipulate that. And with a novel technology, hopefully they will be able to define and distinguish in a better way. Here are some of the examples of cell surface molecules which are involved. These molecules which are expressed on T cells are CD2, LFA1, and ICAM3. And on antigen presenting cells, it is ICAM1, ICAM2, or DC sign. Expression of these adhesion molecules, depends on the phases of interactions. Early on, you will find that CD2 or LFA1 activity is important, but later on, this ICAM3 and DC sign comes into picture to stabilize this complex better. So, as I said, the initial encounter, is mediated through CD2 binding to LFA3, and on the antigen-presenting cells, that synergizes with LFA1 binding to ICAM1 and ICAM2. So this basically complex is important in very early on phase, and this comes into the later on phase. Once the interaction that appears to exclusive for interacting naive T cells with the dendritic cells is that between ICAM3 on the naive T cells and DC sign on the dendritic cells. DC sign is again a marker which we utilize to phenotype dendritic cells, differentiated dendritic cells, and you can use it in flow antibodies are also available for that. DC sign is a C-type lactin that binds to ICAM3 with a high affinity. So here is a simpler cartoon, what we have just discussed, that T cell initially they bind to antigen presenting cells to the low affinity LFA1 or ICAM1. And in this case, basically, your T cell receptor engagement is happening, and your LFA1 and ICAM1 brings those two cells together so that they can interact better, and it will complex and change the conformation so that this junction or this association becomes more and more tight, and cell adhesion molecules are critical in that process. So at this stage, I can get you a tea break because the next set of slides which we have is more discussing about the co-stimulatory molecule. And you all know that co-stimulatory molecules are very important in antigen presentation. And also, these are the molecules which have been exploited in immunotherapeutic protocols. Your CTLA4 antibody, which is a class of co-stimulatory molecules, or PD-1 or PD-L1. Those are all come into this co-stimulatory molecule complex. So we will go through that after a break. Okay? Any question you have so far, what we covered? Yes.
Sir, you said that dendritic cells which are mature, so dendritic cells which the one is the peripheral tissue and the another one is in lymphoid tissue.
No, most of them are in peripheral tissue, and once they pick up the antigen, they go to lymphoid tissue. They don't reside in lymphoid tissue.
Okay.
Yeah, and in lymphoid tissue, once they are there, they will present their antigen to naive T cells, and once the job is done, they disappear, they die through apoptosis.
So, is there any role of dendritic cell in peripheral tissue? Means any type of?
Yeah, so their major role is trying to, survey, it's like a surveillance, they are monitoring, and another job is that as soon as there is some infection or injury, they go to that site of infection and injury. They can migrate there.
Okay. And sir, if they are unable to migrate to lymphoid tissue, sir, does it lead to any type of diseases or means any type of error in the immune system?
Yes, of course, because these dendritic cells, if they cannot go to secondary lymphoid organ, they will not be able to, present their antigen to T cells, which are, the effector cells. So basically, there will be no effective immune response if there is no migration. So you have to understand that there is a division of labor. There are a lot of compartmentalization which is going on. This is all what I am talking about is happening in context with a normal immune response. In cancer, actually, it's very interesting because sometimes cancer itself becomes like a secondary lymphoid tissue. So what you see that these macrophages are dendritic cells which are within cancer or which we can recruit through. Like I was telling you that CCL20, which is a ligand for CCR7, and most of the differentiated dendritic cells express CCR7. So if I take a tumor and inject them intratumorally, this CCL20, I can recruit dendritic cells to tumor itself. And in those tumors, what you will see, and I think we published it almost 10 years ago, that in those tumors, it exactly looks like a secondary lymphoid organ. You see a lot of naive T cells coming to that tumor, and then you can see a massive infiltration of, massive proliferation of antigen-specific T cells. So, and that happens in tumor, that whether that can be recapitulated in other tissues, not possible. The tumor is a pathology, okay? You have to understand. That may be happening in a normal wound, normal wound that is existing there for a long time, not a wound that heals. In that wound that heals, dendritic cells have done their job, they are gone, they are dead, okay? But there's some wound that is not healing. You can see these cells, they go there, they may hang around longer, but you will have aberrant presentation and aberrant activation. And again, cancer is nothing but the wound that never heals. You understand? So, but yes. Cell adhesion molecules, yes. So the question is that the expression of these cell adhesion molecules are tissues. It's
depend on the cell's own
cell specific
own experience, or it's a set pattern which is like determined that this much for this cell adhesion molecule, or a sequential thing.
So, in this case, at least in antigen presenting cells T cell interaction, there is a set of molecules, okay, which have been defined, and most of these are defined experimentally. When you look at overall expression of these cell adhesion molecules, they are everywhere. Okay, you will find ICAM1, ICAM2, ICAM3 in many other tissues. LFA1, LFA2 is present in many other tissues. The only cell adhesion molecule which will be unique in this context will be that DC sign, which is CD205 or something like that. But what happens in this case is that these adhesion molecules, again, I showed you that their activation is sequential, and the goal is to bring these cells together, stabilize the synapse, and then allow these antigen-presenting cells, these T cell receptor complexes interact with this MSC class 2 or class 1 molecule. Okay. Okay. So we are ready for tea break. Okay. Maybe I will say 7 minutes. Okay, everybody is here? Okay. So whatever we have done this morning is just one thing, one signal, that T cell receptor or B cell receptor, not so much B cell receptor, but T cell receptor recognizing antigen or peptide, how they do that, and how they interact with antigen presenting cells. In that picture, the most important element is that it's not just one man show. There is another signal, they call it co-stimulatory signal, which is important in that process. And that is important for activation and effector function. And they call them as a group of molecules called co-stimulatory molecules. And usually these molecules are not expressed on antigen presenting cells. Dendritic cells, they have some low level expression of co-stimulatory molecule, but macrophages and B cells, they don't have co-stimulatory molecule expression. They do get upregulated when antigen is taken up, and when as they are getting processed or maturation is going on, the expression of co-stimulatory molecules goes up. And once you have these two signals, first your antigen presenting cells, antigen complex with T cell receptor, and the second your co-stimulatory molecules interact, then you have the right signal, and then you will have the right kind of cytokines produced, which will basically be important for expansion of your T cells to generate clones which can go and take care of the infection or that particular antigen. I kept saying that these co-stimulatory molecules are not constitutively expressed, and there is a reason for that. And that's the one question I will have at the end of the day, which I asked you yesterday. And think about it, why we use adjuvant whenever we are doing vaccination. Okay, every vaccine we are giving, we are combining it with some adjuvant. Okay, what's the role of adjuvant? Vaccine has the antigen. Okay, so basically they should, and they don't do anything. Okay, if you just take antigen and inject it, it does nothing. So what's the role of adjuvant? Okay, so we will have that discussion, after the break or whenever we get together. So both specific ligand and co-stimulatory molecule signals should be provided by antigen presenting cells and required for the clonal expansion of naive T cells. And here is the simplest cartoon. You have CD4 positive T cells interacting with antigen which is bound to MSC class 2 molecule. You have CD3 complex, and then you have zeta chain right here. So that's downstream signaling, and they call it signal 1. And the co-stimulatory molecule in this context will be your CD28, which is a receptor present on T cells, and B7-1 and B7-2, which is present on antigen presenting cells. These are the most common and widely studied co-stimulatory molecules. So these will be your ligand, these are the receptor. And so in order for this process to happen, both signal 1 and signal 2, both are required. What happens if there is no signal 2? Basically, your CD4 positive T cells at your T cells will recognize, the antigen, but they will not go through this expansion process. Okay, they will not produce the IL-2 and other cytokines which are critical for inducing that proliferation. And if there are not enough cells, there will be no effective immune response. So the best characterized co-stimulatory molecule is again B7-1, B7-2. In terms of CD, they are called CD80 and CD86. You can actually do flow cytometry to look at your antigen presenting cells, and that can tell you whether your antigen presenting cells are expressing it and expressing it at what level. The B7 molecules are homodimeric members of immunoglobulin superfamily that are found exclusively on the surface of antigen presenting cells. and the receptor for B7 on T cell is CD28, which is another member of immunoglobulin superfamily. So here is a list of all the B7 ligands and receptors, and I will just walk you through, I will spend some time here. So as I said, on T cells, you have expression of CD28, and your antigen presenting cells, which could be your dendritic cells, B-induced monocytes, or B cells. They express these B7-1 or B7-2, which will interact with or bound to this CD28. And in this case, you will have enhancement of response, clonal expansion of response. There is a counteraction, or there is an anti of this response, and that is very interesting and important. The same complex actually can bind also to an inhibitor of this interaction, which is called CTLA4, and this response is immunosuppressive. Okay, and this is important in the sense is that this basically signals in a very different way, and this has a higher affinity than this. Okay, this has almost like 100 folds higher affinity than CD28. So what happens is that initially, when your antigen presenting cells are interacting with the T cell, they will have these B7-1, B7-2 bound to CD28. They will get the right signal, and then they will have, start the expansion. But they don't want that to happen constitutively. Okay, they have to stop that signal. Once they got these right signal, T cells have start producing IL-2. Then they don't want these cells to have continuous exposure and stimulation. Immediately, they turn on this CTLA4 system. What will happen is that it will try to stop that activation signal. Okay, because as I said, that aberrant activation is the cause of many other diseases, and responses could be very different. So there is a very fine balance which goes on between CD28 and CTLA4 interaction with these B7-1 and B7-2 ligand. Earlier in the response, you have these B7-1, B7-2 binding to CD28, and then CTLA4 comes as a stop. That okay, now you have enough signal, now you should stop and go and proliferate and do their job. There are other B7 family ligands. Here is ICOS ligand, which binds to ICOS, PDL1 and PDL2, which binds to PD1, and this is a very hot area where there are a lot of antibodies which have been approved for clinical purpose. And there is another one called B7H3, which I still don't know what it binds to, but it is from the same family. And in this case, again, you can see that expression of PDL1 or PDL2 on antigen presenting cells, they are suppressive in nature, and these receptor for PDL1 or PDL2 is PD1, which is present on your T cell, B cells, and activated monocytes. On contrary, this ICOS is very interesting because ICOS ligand, which is produced, expressed on these dendritic cells, B cell monocytes, constitutively and can bind to ICOS and T cells, and this is activation signal. So what they are finding is that in those patients which are getting these antibody to PDL1, PD and PD1, there is upregulation of this ICOS system, this system. So basically, it's not just blocking the immunosuppressive phenotype, but also enhancing, the T cell responses through this ICOS, not so much to the B7-1 CD28. CTLA4 antibody actually was the first, immunotherapeutic antibody which was approved in 2010 for melanoma. And for PD1 and PDL1, there are more than 7 different antibodies which have been approved so far. And all these antibodies are neutralizing these pathways. Basically, is that they are trying to enhance the T cell responses. Because basic idea is that when you look at these tumor-bearing individuals, immune system is there. It's doing its job, but it's just overwhelmed. And in most cases, when you look at least in the T cell compartment and B cell compartment, there is a these T cells or B cells are either exhausted or they are energic. And when these T cells become energic, when these T cells have only one signal, these T cells have, antigen presenting cells and also T cell receptor complex interaction, but no second co-stimulatory molecules, activity, then they become energetic. Then they know what to attack, but they don't know how to do it. Okay, so that's what has been exploited extensively, and it is a huge market. Many of them have been marketed well. Another key point you have to remember is that all these B7 family members are also expressed on other tissues, those which I have put a star here. They are not just restricted to antigen presenting cells, but they are also expressed on other cell types. That's another important reason that in many different tumor types we find expression of these co-stimulatory molecules, which are important in your cytotoxic T cell response, because that's where another thing comes. If there is a cellular exhaustion or there is a blockage or there is a suppression, blocking these PD-L1 or PD-L2, enhancing the ICOS ligand activity promotes the T cell responses or CTL mediated tumor killing. So, the other aspect, when these two cells interact, is that once a naive T cell is activated, it expresses a number of proteins that contributes to sustaining or modifying the co-stimulatory molecule signal that drives clonal expansion and differentiation. Okay? And some of those molecules are like CD40 ligand, which binds to CD40 and antigen presenting cells, 41BB, which binds to its ligand 41BBL, which is expressed on antigen presenting cells. The effect of these receptor ligand interactions are usually bidirectional for both T cells and antigen presenting cells. Therefore, this process is usually referred as T cell APC dialogue. And as I said, that CD28 related protein CTLA4 is a high affinity receptor for your B7-1 or B7-2, and is also induced during this process, which serves as modifying the co-stimulatory signal. And modification here is that basically once this gets engaged with CD28, it means time to stop. This is basically function as a stop signal. So, a little more about CTLA4. It is an inhibitory receptor for B7 molecule. Once it is activated, T cell express increased level of these CTLA4 only in the activated T cells, not in inactivated T cells. Inactivated T cells or naive T cells will have CD28. But once they are activated by their interaction with antigen presenting cells presenting the right antigen, then the CTLA4 gets turned on. CTLA4 has a higher affinity, and that is important to effectively shutting down the proliferative phase of the response. Okay? Otherwise, this proliferation phase keeps going and going and going. This makes activated progeny of naive T cells less sensitive to stimulation by APC and limits the amount of an autocrine IL-2 production. CTLA4 B7 binding is essential for the limiting the proliferative response of activated T cells to the antigen. So, you understand what's the role of CTLA4 in this context. So, it is a good thing, it is required for our normal homeostatic process. In normal immune response, CTLA4 is as important as your CD28. Okay. Here is a simpler cartoon. You have, your naive T cells, which are expressing your T cell receptor, recognize the antigen in context with MSC class 2 molecule, and your CD28 binds to B7-1, and you have, both signals present. Cells will start proliferating or get activated. Once the cells are activated, your CTLA4 expression goes up, and basically it is serving as like a stop signal. Don't proliferate anymore. Job is done. You have enough activated T cells. Go and do your job. The co-stimulatory signal is necessary for synthesis of interleukin 2. Interleukin 2, I don't know how many of you know about what interleukin 2 is. It is definitely, as the number says, this was the second interleukin which was cloned. What is interleukin? You can speak loud little. What is interleukin? It is a cytokine. Okay. Why it is called interleukin? Why? There is a whole family. There are now IL-37 we have. Okay. There are 37 of them. Why we call them interleukin? There is a conventional. There is a convention. Actually, it happens every year. To define whether a particular cytokine is a interleukin or not. What is the distinguishing feature for a cytokine to become interleukin? Anybody? There is a very simple definition. Interleukin. Leukin comes from leukocytes, okay? So so basically these cytokines are produced by a leukocyte type and works on another leukocyte. It has to be interact, bringing these two leukocyte functions together. Okay, so that's why these are interleukins. So all the interleukins actually they have to be produced by a leukocyte, and leukocyte is used. All immune cells come under this leukocyte family. Okay, and it has to have receptor or activity on another leukocyte. Okay, so that's what interleukin. And interleukin 2 was second one, and actually it is very important in T cell proliferation. When you are trying to grow T cell, you have to use interleukin 2 in your culture. They are not very expensive. They are species specific, so you cannot use human interleukin 2 against mouse or mouse interleukin 2 against human when you are doing your experiment. So anyway, interleukin 2 is an important cytokine which is basically important for naive T cell proliferation, and it is produced very early on as when these activation signals are all established. And this IL-2 production depends on the interaction, and so this CD28 ligation actually, which the co-stimulatory molecule ligation actiwates this transcription factor like AP1 and NF-kappa B that increases the transcription of interleukin 2 mRNA many folds. When T cell recognizes a specific antigen in absence of co-stimulation, means where your B7-1, B7-2 or other co-stimulatory molecules or second signal is missing, very little IL-2 is produced, and these T cells, they don't proliferate, and they don't not only they don't proliferate, they are confused, because they know what to attack, but they don't know where to go, and they are not sufficient or enough in number. This basically leads to something called T cell tolerance, where these cells, they know which antigen they can interact with, but they don't have this co-stimulatory molecule signal or second signal, which they call it. These energetic T cells, they don't produce IL-2, which prevents them from proliferation and differentiation. These naive T cells, they recognize self-peptide on the tissue cells that are not activated. Instead, they may be induced state of energy. Some of these energetic T cells, they persist in vivo, and the significance of this retention of these cells actually still we don't know. It is presumed that these energetic T cells maybe will get the second signal at the right moment, and they will become activated, and they don't have to go through the whole process of antigen, antigen presentation. So these energetic T cells sometimes, they are not classical memory T cells, but they are presumed memory T cells because they already know which antigen they have to recognize, and all they need is the second signal, or all they need is sufficient interleukin-2 level, which can turn them on and can go on to do their job. So actually, interleukin-2 was the first, no, actually not the first. I think it will be the second cytokine which was used as a therapeutics in cancer patient. So interleukin-2 as such, were injected into cancer patient to enhance the immune response. And one of the idea is that the level of interleukin-2 in these patients are low, and hopefully if they can give them enough. The first one was interferon alpha. Interferon alpha was the first cytokine which was used as a therapeutics in cancer, and interferon basically does multiple things, interferon alpha and beta also. Interferon alpha is not just immunomodulatory, but also it is anti-proliferative, so it can directly also inhibit the cell proliferation. So here is in a cartoon way, you have antigen presenting cells, just co-stimulatory molecule engagement will have no effective T cell response because this antigen T cell receptor recognition is important. Or if you have just a specific signal where your T cell is interacting through your T cell receptor or MSC class 2 peptide complex, will not be sufficient. So lack of either one of those signals probably leads to inactivation of T cells. This is called T cell energy, where you have these antigen recognition, but no secondary co-stimulation. In the case of only secondary co-stimulation, you will have no response at all, actually, because there is no antigen recognition. So the requirement for one cell to deliver both antigen specific signal and the co-stimulatory signal is crucial for preventing immune response to self antigen. Okay, this is another important message. You have to remember that both of these signals, signal 1 and signal 2, has to come from the same signal, the same cells. So T cells and antigen presenting cells. One T cell and one antigen presenting cells both have to give these both signals together. It's not like one T cell binding to antigen presenting cells, getting recognized this antigen T cell receptor complex, and another antigen presenting cell giving them these co-stimulatory signal. That will not be sufficient, and that will not be effective. Otherwise, we will have immune response everywhere. They are there. So that is also one of the limiting step in this response. Here are some interesting examples. Naive T cells stimulated by virally infected dendritic cells, and the recognition is happening here. That your both signals are present, signal 1 and signal 2, and these cells basically are armed and ready to do their job. And in this case, these cells basically, will be your CD8 positive T cell, and these cells basically will be responsible for clearing those virally infected cells. So once they are activated, they will go and bind to whichever cells which are expressing these particular antigen. They will recognize them, get engaged with them, and they will kill them through the pathways which we discussed on day 1. Okay. On the other hand, when a naive T cells, they recognize self-antigen and epithelial cells, they basically will not get the right signal. These antigen-specific signals alone, it can induce energy, and it will make those T cells unresponsive. And this is what happens actually in context with your self-antigen, because these T cells basically, even though if they are binding and recognizing these, they will not be able to get the right signal, right co-stimulatory molecule signal. In this case, actually, if those dendritic cells are in the vicinity, see they have these co-stimulatory molecules. If they just recognize this, and this dendritic cells comes here and gives the second signal, that will not work out. Exactly, that's what the statement which I was making. Otherwise, these cells can also become activated and start killing those self-antigen expressing cells. Once these effector cells are armed, they don't need further co-stimulation. That's interesting concept. Once these cells are stimulated through your signal 1 and signal 2, they start producing IL-2, they start proliferating, and these proliferated cells, once they differentiate, they can go and do their effector function, and in this context, it's mostly CD8 positive T cells, cytotoxic killing, and so on. And this is where you might be able to get some of the answers, the questions which I was asking you guys. Microbial substance can induce co-stimulatory activity in macrophages, and that's exactly what the concept of adjuvant is all about. In macrophages, these cells that don't constitutively express co-stimulatory molecules, even they don't constitutively, they express very low level of MSC class 2. So, once they take these bacterial and non-bacterial antigen or bacterial antigen, they upregulate these MSC molecules or co-stimulatory molecules, and then they become really fully active. So, if they are getting just like a non-bacterial protein antigen, which is just antigen in itself, okay, and in that context, these unstimulated macrophages, they do not deliver a co-stimulatory signal to T cell, and in that context, when the second signal is missing, that will create energetic T cells. So, if you just take the antigen in itself, and if you inject or immunize in some individual, this is exactly what will happen. At that stage, antigen is mostly its peptide sequences which you are going to use, or a product from some pathogens. If it is a bacteria, what happens is that once these bacteria gets phagocytosed by macrophages, which they are very good at it upregulates co-stimulatory molecule. Your now macrophages and not only have MSC class 2 upregulation, but also have this second signal of co-stimulatory molecule expression, and then you will have a nice proliferation and differentiation. And many of these macrophages are the one which gets to the site of vaccination, uptake not just the antigen, but also they will take out some of those bacterial products. They will process antigen, they will upregulate these co-stimulatory molecules and exactly do what they are supposed to do. So, this is what happens exactly when you combine your antigen of interest with a bacterial product, and those bacterial products serve as an adjuvant. And in this context, exactly what happens, as I mentioned, that this will upregulate a co-stimulatory molecule, enhance the antigen presentation, and you will have a very strong immune response. So, in many ways, basically all you want to do, why you want to use adjuvant? You want to use adjuvant to not only enhance the antigen processing and presentation, but most important, to upregulate the co-stimulatory molecule or second signal. Okay. There is another twist to this whole thing. Still, it's not end of the story. The adjuvants do little more than this. But at a biological level, this is important. There is another aspect of adjuvant which we discussed yesterday. Actually, if you think and remember, whenever you use adjuvant, what do they do? So, most of these adjuvants, as I said, they will be derived from bacterial product, okay? And those bacterial product will have what? PAMP, right? What they will do, that they will engage, toll-like receptors. Also, that engagement and involvement of that inflammation, keep that in mind, that these macrophages, they reach there because there is inflammation, and that inflammation is induced in many ways by those adjuvants which is used. So, adjuvants are inducing inflammation, enhancing expression of co-stimulatory molecules, and MHC class 2 expression and antigen presenting cells. Okay. So I think our discussion is over on adjuvants, right? Microbial substance can be used, can induce co-stimulatory molecule in macrophages, and that exactly I think I have talked a lot. I showed you the slide, and this is what I have put in words. But more important is that when bacteria are mixed with the protein antigen are rendered immunogenic. This is a very simplified statement, because bacteria induces co-stimulatory molecule in the antigen presenting cell. Such added bacteria can act as adjuvant. Okay. Activated T cells, they synthesize IL-2 and IL-2 receptor. I am not going to go in detail about IL-2 and what IL-2 does, but it is important to remember that once this whole antigen presentation, activation all happens, this is the molecule which comes into the picture to induce proliferation of those T cells to generate effector T cells. So here is a simple cartoon. Low resting T cells, they express only a moderate affinity IL-2 receptor, but once they get activated, they start expressing this high affinity IL-2 receptor. And when IL-2 is in the surrounding, these receptors get activated and induce T cell proliferation. What happens to those T cells once the job is done? You don't want them to be hanging around in the system. What happens to them? They die. Question is, where do they die? They just don't die everywhere, right? They die in a very specific organ, actually. What is the graveyard of all these immune cells? Spleen. Spleen is a graveyard, actually. So many of them will get cleared in a spleen. Co-stimulatory molecules are also involved in CD8 T cells. Naive CD8 positive T cells require more co-stimulatory activity to become effector antigen presenting cells. And these, whatever we are calling it as high co-stimulatory activity or low co-stimulatory activity, will all be defined by the type of antigens which they are exposed to. In case of viruses, it's always high stimulatory. Okay, viral response is very rapid, and in all the cases, we have always high co-stimulatory activity, and in all of those cases, CD8 positive T cells responses are required. Whenever it is low, they do get CD4 positive T cell help to promote or enhance that activity. Naive T cell, CD8 positive T cells can be activated directly by potent antigen presenting cells, and here is your virally infected cells. Virally infected dendritic cells, not any cells, and in that context, basically, these CD8 positive T cells, they are recognizing the antigen in MSC class 1 context. Your co-stimulatory molecules are there. These CD8 positive T cells will start producing IL-2 and proliferate and create, more CD8 positive T cells, differentiate and go and kill the virally infected cells, which have the particular antigen, whatever antigen they are getting presented through these virally infected dendritic cells. Some CD8 positive T cell responses require CD4 positive T cells, and in this context, both of these has to be from the same cell type. So your antigen presenting cells, like your macrophages, are dendritic cells. They have both MSC class 1 and MSC class 2, and they will, basically. And in these cases, these are not mostly high affinity. These are low affinity or moderate affinity antigens, which will be recognized by your CD8 positive T cells. But it will not induce enough, response, or it will not generate your IL-2 production. But the same thing is also presented in MSC class 2 contact, and this MSC class 2 basically will bring these CD4 positive T cells, will have the right second signal, will start producing your IL-2, can be utilized by CD8 positive T cells, which will be basically will go and do their effector function. So this is what exactly here says, that that IL-2 production signal comes through this CD4 positive T cell engagement with your antigen presenting cells, and the recognition is happening, but there is no engagement of the second signal in context with the CD8 positive. Later on, it does get upregulated the other ligands like 4-1BB or 4-1BB ligand, and that enhances the overall response. So sometimes CD8 positive T cells, they need CD4 positive T cell help to get the job done, and in those cases, most often that will be your low affinity or moderate affinity immune response. Okay, so the question, I think you may all know the answer by this time. What happens to dendritic cells following antigen presentation? They need to go and die. You know, that's a simple answer. And it is important also because you don't want those antigen presenting cells to hang around. Do immature dendritic cells directly tolerize T cells? And answer, you will have to find out, and we can talk about it. What happens if you recruit a lot of immature dendritic cells? And actually that's what we do if we do most of these, you know, injecting cytokines or chemokines to recruit dendritic cells to the site of tumor, which I was mentioning earlier. Most of these dendritic cells will be immature, and they, rather than enhancing the T cell response, they make them tolerized. Some discussion on what happens to B cells, and in this context, because B cells are also important. One thing is very important is that whenever B cell activation is involved, there is always T cell help, you know, unless that is a super antigen. Okay, in most of the antigenic response where the B cell is involved, it always needs T cell help. And T cell help, when you talk about, it is always CD4 positive T cell. If it is super antigen, they don't need any help. Okay, and I hope you all know about super antigens. You know, they can directly engage these B cell receptors and activate these B cells to cause the massive pathology. And often that's why they call it linked recognition. This means that before B cells can induce to make antibody to infecting agent, a CD4 positive T cell is specific for peptide from this pathogen must be activated to produce appropriate effector response. So there has to be relatedness in that antigen. So B cell basically will recognize that antigen because that antigen will bind to B cell receptor directly. And also that antigen has to be, you know, processed and presented to CD4 positive T cells, okay? And those CD4 positive T cells have to know exactly that's antigen they are talking about, or B cell, and then they will be actually, then they will have the effector function. The two cells need to recognize identical epitope. That's another important, feature. The T cells can recognize internal peptide that are quite distinct from surface epitope on the same protein, but they have to be from the same protein, which is recognized by the B cell. And here is a simple cartoon, showing that B cells basically they bind to that, and this is like your pathogen, which will bind through the specific epitope through the B cell receptor. This epitope binding can cause the internalization of this bacteria or pathogen, whatever it will be, and then there will be processing and presentation of the antigen from this pathogen on the surface of B cell in contact with MSC class 2 molecule. So, and that basically will cause the production of factors or cytokines, which will be utilized by the B cell, which have recognized these pathogens. And that B cell will go through the process of differentiation, become plasma cells, and start producing antibody. Simplified version, okay? But there are a lot of else happening in between. But all here you need to remember is that, CD4 help is required for these B cells to become activated become plasma cells and start producing antibodies. That's one point. The other point is that recognition, that of course initial recognition is that antigen which is on the pathogen is getting recognized on B cells. But that antigen has to be presented and processed by these cells to get T cell help, which is CD4 positive T cell help, to start producing cytokines, which will be important for B cell maturation, differentiation, and transformation into plasma cells. What are the B cell growth factors? That's the question, okay? Over there on T cell, I told you interleukin 2, interleukin 2, interleukin 2. What is B cell growth factor? What growth factor is required for B cell? Okay, during lunch, okay, you go to Dr. Google or whoever is your point of interest, okay, and find out what growth factor is required in this, okay? That's your take home. IL-6. IL-6 is also known as B cell growth factor. Which one? IL-7? IL-6 was a correct answer to some extent, okay? Because IL-6 is also known as B cell growth factor. It was originally isolated. When they were isolated it, they called it a B cell growth factor, okay? So you are right, it's IL-6, okay? But there are other growth factors which are cytokines, not growth factor, other cytokines which are produced. What other cytokines are associated with humoral response, which is produced by CD4 positive T cell help. Okay. Now we are going back. Okay, this is a good exercise. So IL-6 is B cell growth factor. Okay, but this cytokine is coming from your CD4 positive T cell. Okay, which is causing antibody B cell differentiation and antibody production. So you have many types of T helper cell, right? You know Th1, Th2, T helper 1 and T helper 2. T helper 1 causes a cytotoxic T cell response, right? It's a cell mediated immune response which is mediated through cytotoxic T cell. Th2 is your antibody mediated humoral response. So what cytokine is associated with Th2 response? Th2 type 2 helper cells, what cytokines they produce? This will be your type 2 helper cell, okay? T helper 2. What cytokines they produce? There will be no lunch break if you don't give us the answer. Huh? It will be immunosuppressive. Okay, I have to eat lunch too, right? Okay. So it is IL-4 and IL-10, IL-13. Remember? Do you recall? IL-4, IL-10, IL-13. So if I have to pick, it will be either IL-4 or IL-10, okay? IL-6 also is upregulated in this context because it's a directly B cell growth factor. So the specific recognition of B cells by T cells sensitized to the same antigen or pathogen depends on ability of antigen-specific B cells to concentrate the appropriate peptide in its MSC class II molecule. It has to be presented in context with MSC class II. B cells that bind to a particular antigen are up to 10,000 times more efficient at displaying the peptide fragment of that antigen on their MSC class II molecule than the B cell that do not bind to that antigen. So that gives the specificity. Armed helper T cells will help to those B cells whose receptor binds to antigen containing the peptide they recognize. That's the dual recognition or linked recognition which we are talking about. The requirement of link recognition has important consequences for the regulation and manipulation of humoral immune response. One of that is that this linked recognition helps to ensure self-tolerance. In everywhere in immune response, they want to make sure that your immune cells should not start attacking your own cell. Okay. Whether it's T cell response, B cell response, even in macrophages, they have those kind of signals which are important. So, always trying to induce self-tolerance. So, again, I think this will be interesting not just for academic purpose, but also for your own research. Dendritic cells, macrophages, and B cells, they differ in their selectivity of antigen uptake, their antigen processing properties, and their co-stimulatory and migratory behavior. Thus, have a distinct function in initiating T cell response. So, depending on what is your antigen, what area you are working on, okay, you will choose and work with different kind of antigen presenting cells, choose and decide on different kind of adjuvants and factors which will help stimulate or suppress the immune response. Or even for basic understanding, you know, the response against a specific pathogen will vary depending on what type of pathogen you are or infectious agent you are interacting with. Okay? And antigen presenting cells play an important role in that. So, there is a very nice division of labor and you know, it's I will say that it is basically creating a whole different kind of complexity within our immune response. And most of these actually complexities are created to put checks and balances, you know, at every step. They just want to make sure that our immune system is exactly going to work against the threat, not against everything which we ever encounter to. Okay. So, thank you very much for your attention, and we'll continue our discussion. Any question?
हेलो एक थप्पड़ खाएगा यहाँ पे माइक है Sir, what is the significance of interaction between tissue resident DC and ECM in any kind of inflammation? Dendritic cells and extracellular matrix of the cells in that tissue. What is the significance?